Oxidation of glucose, or glycogen, to pyruvate and lactate to produce energy, in the form of ATP, is called glycolysis. This was described by Emden, Meyerhoff and Parnasse. Hence, it is also called as Emden-Meyerhoff pathway. It is a catabolic pathway. It occurs in cytosol and virtually in all the tissues. This pathway is unique in the sense that it can utilize oxygen, if available, which is called aerobic glycolysis, and it can function, in the absence of oxygen also, which is called as anaerobic glycolysis. Enzymes involved in glycolysis are extramitochondrial. Significance of glycolysis. Glycolysis provides energy to the body, especially brain. Among carbohydrates, brain utilizes only glucose as a primary source of energy. It is also the main pathway for the metabolism of other hexoses, fructose and galactose. It produces pyruvate, which then provides different metabolic intermediates. Erythrocytes and nervous tissues derive its energy mainly from glycolysis. It is the main pathway of carbohydrate metabolism to generate energy, i.e. ATPs. It produces ADP, NADH2 and pyruvate. There are two types of glycolysis, that is aerobic and anaerobic glycolysis. Aerobic glycolysis takes place in the presence of oxygen, while anaerobic glycolysis takes place in the absence of oxygen. End product of aerobic glycolysis is pyruvate, while end product of anaerobic glycolysis is lactate. In aerobic glycolysis, NADH2 is produced, which gives 3 ADPs, through respiratory chain, which works only in the presence of oxygen. In anaerobic glycolysis, respiratory chain stops, in the absence of oxygen. Therefore, the NADH2 produced in anaerobic glycolysis cannot give 3 ADPs, instead it reduces pyruvate to lactate. Total 10 ADPs are formed. In aerobic glycolysis, 2 ADPs are utilized, and the net gain is 8 ADPs. In anaerobic glycolysis, total 4 ADPs are formed, 2 ADPs are utilized, and the net gain is 2 ADPs. Difference between glucokinase and hexokinase is as follows. Glucokinase is only present in liver, while hexokinase is present in liver as well as in other body tissues. Activity of glucokinase depends upon the nutritional state of glucose, that is, it becomes active only when sufficient quantity of glucose is present. While the activity of hexokinase does not depend upon the nutritional state of glucose, that is, it remains active even at low glucose concentration. Glucokinase has high Km value, while hexokinase has low Km value. Glucokinase is not an allosteric enzyme, while hexokinase is an allosteric enzyme.